Okay, getting into chapter 4. So first of all, let's reintroduce this idea of what bacteria and archaea are. They are not eukaryotes, which is true nucleus. They are akaryote. Another term for that would be prokaryote. Those are interchangeable. Okay. So bacteria and archaea, they do not have a nucleus. This is the key defining characteristic of the difference between bacteria and archaea as akaryotes versus the eukaryotes like us. They don't have a nucleus. They actually don't have any membrane-bound structures inside of their cells. They have a membrane that encloses them. That's what defines a cell, having that membrane that encloses everything, as well as being able to multiply that cell. But they don't have any other structures that are contained within a membrane inside of them. They don't have a nucleus. They don't have mitochondria or endoplasmic reticulum or a Golgi apparatus or any of that. No chloroplasts, none of that. So it's really important key difference between them and our typical eukaryotes, which we'll get into in chapter 5. So here's our basic bacterial cell structure. I'm not going to go over this figure here in the lecture, but I'm just pointing this out as a great figure to come back to when you guys are studying and wanting to understand the different parts of the bacteria and what their functions are. We're going to go over a good number of these and not all of them. There's some of these that we're not going to talk about, like inclusions and granules. These we're not going to talk about, or microcompartments. We're not going to talk about those. For the ones that we do talk about, be sure you understand them to the degree that we talk about them. All right. So bacteria and archaea both, they are single-celled organisms. They're always single-celled. They're never multicellular organisms. They can act as a group and they can interact with one another as a group, but they are not multicellular. You can still take one cell and that cell can exist by itself and reproduce by itself, and each of those progeny can do the same. These are single-cell organisms. That's not the case with you and I. If I take one of your cells, it's not going to grow up into a whole new you on its own, right? So these guys can. And so they're single-cell organisms. Each cell is capable of reproduction. It has its own metabolism. So these are creating molecules and breaking down molecules, and how we create energy in the cell. That's metabolism. All of the entire chemical processes in a cell, that's metabolism. Okay. Nutrient processing, being able to take in whatever nutrients you need and use them for building new cell parts and creating energy for the cell, all of that. Each cell can do all of those things. When we act together as a group as bacteria, this could be, and we've already introduced the concept of what a colony is, one cell that's just divided and built up on itself in a big old pile. So that unit kind of can interact with itself like that in that big old pile. But we also have biofilms, which we'll talk about what those are in just a moment. And then nanowires, which is sort of like lines or long groupings of bacteria of same species or different species, and they can kind of send, I don't want to say signals along the way, but like chemical interactions along the way. We're not really going to ever talk about those, and I'm not going to test you about it, but it is something that exists. We are going to talk about, we've already talked about colonies, but we're definitely going to talk about biofilms. So moving on, we have different shapes, sizes, and ways that cells can arrange with each other when we're talking about these single cell conglomerates that can exist. The three general shapes of our bacterial cells: coccus, bacillus, and curved. So if it's not a coccus and it's not a bacillus, it's probably going to fall into curved. And there's a lot of different kind of groups in that curved. Vibrio, which is comma-shaped. Spirillum, corkscrew, and spirochete, also, they obviously have spiral aspect to them. And I'm not going to get into much detail with the curved groups. And I usually don't test much about them, okay? But you definitely need to know the difference between a coccus and a bacillus. In fact, these terms, you've probably already heard them, right? You guys have already heard coccus. Like most people know Staphylococcus aureus. Its cell shape is in its name. So already you know that Staphylococcus aureus, its cell shape is round, spherical. It tells you in its name. So there's that. Some organisms contain their shape and or their arrangement in their name. Not all of them do, obviously. If we're talking about Escherichia coli, E. coli, that Escherichia doesn't tell you anything about its shape or its arrangement, which we'll talk about arrangements in a moment. You could also have a pleomorphic shape. Pleo means many, and morph means shape. So they could have just, they don't have a defined shape. So you have them growing and they're all the same species, but some of them are roundish, some of them will have curve, some just have like a amorphous shape or whatever. And we'll talk about why that happens in some cells in a little bit. But our two biggies that you really need to know the term for and understand the difference between are our coccus and our bacillus. In the lab, all of the organisms that we have are either coccus or bacillus. I have one species that is coccobacillus. So it kind of falls into like a pill shape oval. We'll call it oval, where it's a little bit off from the typical round of what we see with a coccus or the typical rod of what we see with the bacillus. I'll show you some pictures in a moment of these. Yes.
So the bean shape, would that be like yeast is like shaped like a it looks almost like a soup plant, would that fall under?
So yeast typically have a round shape, but yeast are not bacteria. They are eukaryotes. So this is what we're talking about really, which is the bacteria for now. Yeah, that's a good question. And if I'm testing you guys over this stuff, I'm not gonna make it vague. I'm gonna show you a picture of a guy that is round or a picture of a guy that clearly is like rod shaped. And I'm gonna show you some examples of those. Here we go. So here we go, have our coccus. And I know they are showing you sort of like vague in between kind of shapes. Like look at this guy and look at this guy. Come on now, right? But what I need you guys to tell me the difference of coccus to you, round. Bacillus, the official name for it is bacillus, is rod. And for you guys, that's the difference. That's what you need to know. Coccus round, bacillus rod. Now, cocci, look at C-O-C-C. Those three very round letters should remind you that it's round, okay? Bacillus, when we're thinking of that word, has, like here we have a bacillus down here in this term. The two L's, think of that, there we go, rod, okay? And when we see pictures of these guys, look at our coccus here. These cocci are clearly like kind of in weird random bunches, right? And these cocci here are attached to each other in long chains. And we have a couple of chains or three different chains kind of going on here. So the round cells can arrange in different ways. We'll talk about the arrangements in a moment. But you can see the cells individually are clearly round. So each of these is a cell, very spherical in shape, coccus. With our rods, our bacilli, our rod shaped, long rod. And the ones that I will test you over if I give you a picture, I don't give you pictures on the lecture exams, okay? But on the lab exams, you guys are gonna be looking in a microscope, and you're gonna have to tell me if what you're looking at in the microscope is coccus or bacilli. And they're gonna be obvious. They're not gonna be whatever this BS is here, okay? Not that, we're gonna go for obvious, okay? So be able to recognize those differences for sure in the lab. Definition wise, know that coccus is round. So if I describe to you guys in a question that the lab reports to you that they saw cocci, what shape of cell would that be? Round or spherical, okay? So you would know that automatically. Right, so that's the idea of those. And again, I'm not really gonna test you much over the curve. There are things that exist and we'll talk about some bacteria that have curved shapes, but specifically those shapes, I don't usually test over. I want you to focus on the coccus and the bacilli. Okay, so let's talk about how those cells can be arranged. Cocci, they have their certain kinds of arrangements, and the bacilli, they have certain kinds of arrangements. And some of them overlap the definitions. All right, so diplo means two, di, right? So diplococci, we come in pairs of cocci. One very infamous example of a diplococcus would be Neisseria gonorrhea, as well as all the Neisseria species. So Neisseria gonorrhea, obviously the cause of gonorrhea, and then Neisseria meningitidis, one of the possible causes of bacterial meningitis. If you, and we'll talk a little bit about meningitis here in a little bit, because this is actually one of those things that's very, very easy to diagnose with just a gram stain. And some people are like, oh, gram stain, like, what are you actually going to be doing that in medicine? Man, if your patient comes in with meningitis, that is the first test they're going to do, is a gram stain. The patient comes in, they have sensitivity to light, they have stiffness in their neck, a headache, strong fever, maybe they're a little disoriented already and things like that. They come into the ER, the first thing you're going to do is prescribe broad spectrum antibiotics. So there's a lot of different things that can cause meningitis. So meningitis literally just means you have light sensitivity, you have neck stiffness, you have a fever, and you have a headache, and those things that are associated with symptoms of meningitis. It's something that we call a syndrome. It's defined by the symptoms and the signs that we see associated with the illness, not by the cause. Whereas the flu, influenza always causes the flu. You know the cause of it. It could be different strains of the flu, but it's always influenza. Not the case with meningitis. Meningitis can be caused by bacteria, and there's a lot of different strains of bacteria and different species of bacteria that can cause it. It can be caused by viruses, and it can be caused by fungus. So your patient comes in, they have meningitis. What's causing it? Step one, I don't care. Let's give them antibiotics because the bacterial meningitis is so severe. You don't have time to play around with that, especially if we're talking about a little kid. We don't have time. It's causing damage. We're talking permanent hearing loss, paralysis, coma, things like that. We don't play. So we give antibiotics, broad spectrum, immediately. The second thing we're going to do, we're going to get a spinal tap and collect cerebrospinal fluid. And what are we going to do with that? Send it to the lab and order a gram stain. Because that gram stain, which we're going to learn about in just a moment, will tell you everything you need to know for whatever could possibly be causing it. All right. And we'll talk about how that will work and what it will tell you in a little bit. But even if you look in your gram stain and you see gram-negative diplococci, it is Neisseria meningitis. Hands down, that is what it is. It couldn't be anything else. That is like one of the major culprits of it. It's not as common as other types. And again, we're going to come back to this and get into more detail in it in a little bit. But already Diplococcus is telling you something. We haven't even gotten to the gram stain part and what that means. Diplococcus already tells you something. Other causes of meningitis, they don't have that. For example, the most common cause of bacterial meningitis is Staphylococcus pneumoniae. We call it the pneumococcus. Yeah, it's the #1 cause of pneumonia as well. But it's also the #1 cause of community-acquired meningitis, bacterial meningitis. So if you looked at a simple stain, not even a gram stain, but just a simple stain, Staphylococcus versus Diplococcus. It's already up here on the slide. Staphylococcus, we have those irregular clusters. So let's go back to that picture we were just looking at. Staphylo, irregular clusters. Now it'd be very easy to tell the difference between random two pairs. It looks like little butts all over the slide. It really does. The little butts all over your slide versus the random. Already, we have a way that we can tell the difference. And you better hope that if you had to choose between the two, between the pneumococcus and the meningococcus, you better hope it's the pneumococcus. And it is still very deadly, especially to little ones. Okay, so we have there our groupings already. Diplococcus is pairs. Tetra always means 4, so groups of 4. Staphylococcus, these are our regular clusters. I'm never going to ask about micrococci. Streptococci, these are chains. And in Sarcina, you will literally see like cube-like shapes. They look very cubic. You're almost never going to see them. You'll never see them in the lab. The only ones I'm going to show you in the lab will be Strepto or Staphylo, or no arrangement, because that's a possibility too. Sometimes they don't have an arrangement. But if they do, these are the ones they would fall under. Right, so these are our cocci. These are our round ones. For our bacilli, which are our rods, we can still have diplo. And they're usually going to be pairs, just like end to end, like this. Okay, end to end. Strepto, now just longer chains. And then palisades. So what's a palisade? A palisade, they're like fence. So they're right next to each other, like fence posts. So obviously only bacilli would be able to do this. You'd have to have that rod shape in order to accomplish being able to tell the difference between end to end versus side to side. So palisades, and it's actually a term that we use in like architecture and things like that for fences, for barriers. Just like fences. I don't really care too much about how the spiral shaped ones arrange themselves. I won't test you on that. All right, let me delete that. Next slide. All right, biofilm. So what's a biofilm? The best example I can give you of a biofilm, have an everyday sort of exposure that we might have for biofilms, will be the plaque on your teeth. And yeah, you've got plaque on your teeth right now. Even if you brush your teeth, even if you chew gum, even if you use mouthwash and floss, you've got some building up always. If it's been more than an hour since you brushed your teeth, you probably would have already thousands that have already multiplied on your teeth, giving you a film. Just from your normal flora that's just growing. And what they'll do is bacteria will come in and stick to rough surfaces. Typically, it's a great spot for them to stick to. And they have sticky coatings that allow for that. And then other bacteria can come stick to them. They could be different species or the same species, and then we'll multiply in that whole setup. We have this conglomerate of bacteria, of archaea, of protozoa, of other eukaryotes like yeast, can all get involved in these different layerings of cells. So these are microbial kind of mini ecosystems that can happen from these guys like just sticking up onto each other and building off of each other. It's usually multiple species involved. When we are forming our biofilms, again, you usually have some sort of surface that these guys will stick to, and then they have this sticky coating that they can develop that helps with that sticking. It helps them stick, and it helps other bacteria stick to them. And then we have this buildup of this little ecosystem, creating that film. And then these guys within that film can secrete proteins or other molecules, or even just the waste that they make. It lets the other microbes around them know about the health of the entire biofilm. So if you start sensing that there's a lot of waste or byproduct that you only see when organisms are dying, you might not want to be a part of that biofilm anymore. But if they're also making waste products that are pretty common for happy, like if you go to a party and you see like there's a ton of like beer cans hanging around, and there's like, you know, food trays, people are eating out, like, this is a pretty good party, people are having a good time. But if you go to a party and you just see that, like, you know, there's cigarette smoke everywhere, maybe not your kind of style of party that you don't want to participate in. So that's that sort of stuff. So they can pick up on the cues of the bacteria that are living in the biofilm based on waste products and what they're leaving around, as well as just chemical signals that they can send out. This is the way of them to be able to communicate with each other. That's called quorum sensing. Yes.
So the biofilm is the like habitat itself.
Yeah, sort of. Yeah. So it's like the entire, like the literal film that would be forming as a result of all these microbes living together. Yeah. So, so within that sort of environment, they can communicate with each other through quorum sensing, and that's just their ability to recognize the chemical signals from the guys around them and read the room, if you will, on how you know great of a situation that is. Quorum, if you don't know what that word means, is basically like something that like the Senate and things like that would be using to say if we have quorum, it means we have enough people to actually do business. So quorum is like a grouping of people, typically. So it's a grouping of bacteria and other organisms communicating. All right, bacteria and archaea, and particularly bacteria, because most of what we're talking about is bacteria here. They have appendages. Some of those appendages, let me hide this. Those appendages can be for motility, so for moving around, or for adhesion, or even for moving things between different cells, okay? So the only appendage that bacteria have for moving, they only have one that they can use for moving. Not all bacteria can move. There are bacterial species that are non-motile. So Staphylococcus aureus, which if we take a step back talking about our shapes and arrangements, Staphylococcus aureus, it tells you in its name, staphylo, it's irregular clusters or grape-like clusters, however you want to look at that. Coccus, round cells. So we have irregular clusters of round cells, it tells you in its name. Staphylococcus aureus is non-motile, so it does not have flagella. Okay. I don't need to know that from its name, but I just happen to know that and I'm telling you that. Whereas when we look at E. coli, it has more than one flagella. So flagella, motility, okay? And then we'll talk about the other guys in a moment. We'll start with the flagella. One flagella is called a flagellum, okay? But I usually just am only gonna say flagella because I'm not very particular about that. Sort of like the media thing, like if you want to talk about one, I usually let's just say media. But just, you know, for semantics reasons, flagellum is a single one. Multiple is flagella. And these guys are protein-based structures with certain like protein components that are all going to work together to allow for this long little tail guy to rotate. It does not move in a whip-like fashion. It doesn't go like this. It only rotates. It rotates either clockwise or counterclockwise. That's really important because when we start talking about our like eukaryotes and how they get around, because we do have single cell eukaryotes, okay? And some of those single cell guys need to be able to get around, and they might have flagella, but their flagella can move in a whip-like fashion, and they have a membrane that surrounds their entire flagella And their flagella are like way more complicated than these guys. But these guys, they work. It works well enough for them. And so this will rotate one way or the other. And if I were to show you an electron micrograph, which can show you down to like an actual atom, or to take a picture of this using an electron microscope, you would actually see that it does in fact look like that. It looks like a motor almost. Pretty impressive. This will rotate clockwise or counterclockwise. If you have flagella, you can have specific arrangements of those flagella around your cell. This could be polar or it could be non-polar. So the polar ones, you have flagella at one or both ends. And then we have the non-polar, which is peritrichous, which is this one down here at the bottom. Peritrichous just means you're random all over the surface of the cell. So you're not at one end or the other, but you're all over. Okay, so that's peritrichous. Trich or trick means hair. Always what it means, even if we're talking about trichotillomania, that's people who pull their hair out compulsively. So trich means hair. So monotrichous means they have one hair, one flagella. Mono, one. So this picture in A depicts that. Next we have lophotrichous. This is tufts. So in picture C, we have a pretty good picture of that, sort of how E. coli would look. They have a tuft at one end. Amphitrichous, you have usually one flagella at each end. I don't have a good picture of that one on here, but it's usually one flagella at each end. And then we have the peritrichous is all over. So obviously B, you can see this guy here, very good example of a peritrichous arrangement of flagella. Now, clearly, if you have just one flagella, you might still be motile, but if you have multiple, they can control them individually. So if you have multiple, now we're talking about if you can only rotate it one way or the other, but you have multiple all over your cell, you're going to have a lot better control over how you're moving than if you have just one flagellum. So that's another thing to consider with these guys. So why do these guys move? How do they move? How are they responding to things? They don't have a brain. They're not thinking about things consciously. They're just responding to chemicals or light in their environment. Some of these guys, they're going to be like our cyanobacteria, the ones that built our oxygenated atmosphere back in the day. Those guys are photosynthetic. So they might want to be near light. So if they get any sort of twinge, oh, there might be light near, then they might want to move more towards the light. Chemotaxis, on the other hand, is just responding to chemicals in your environment. And in fact, this is something that we can actually respond to. And I'm not just talking about pheromones or something. I'm talking about if I bake cookies fresh and I bring them in here and you smell them, you might see that as a positive, smell that you might want to move towards. And you'd be thinking about that probably and react to it that way. Bacteria can pick up on chemicals from their environment and want to move towards them if it's favorable, like that. So that's positive chemotaxis, moving towards a nutrient or something like that beneficial to it. However, negative chemotaxis, if I put a fresh pile of dog poop up here, who's going to want to be anywhere near that? So that's negative reaction. Again, with the smell that you would get from that, that's chemicals reacting with your cells. And they're going to react to things that are negative to them as well. If they get any sense of negative toxic products or waste things or anything that could signal something dangerous for them, that their cells can pick up on little minute levels of that and they might want to move away from it. So that's negative chemotaxis, just reacting to chemicals in their environment. All right, so we have only the ability to rotate those guys clockwise and counterclockwise. So let's say you are spinning your flagella in the counterclockwise motion. And so you're spinning counterclockwise, and you're kind of going, then suddenly you switch to clockwise. You're going, should switch, just like if you were driving straight in your car in drive, and you suddenly switched it into reverse if you're driving a manual. Yeah, your car would not thank you for it, but it would probably shudder a little bit, and then you'd be going in the other direction. And so they'll kind of do this randomly, and they're not governed by four wheels, right? They're kind of just living in whatever environment they're in. And so they could just switch between those two, and they will just do that randomly just to get a feel. It kind of lets them kind of perceive their environment randomly. However, once they start getting a signal or pick up on a chemical in their environment that is positive or negative, now we might be running and tumbling more frequently or running longer and tumbling less if we start picking up on more or less positive signal in whatever direction we're going. So usually the movement can be relatively random and we can't entirely control the direction we're going, but we're just going to run and tumble and try to control it by just switching between running and then tumble. Tumble is just when you switch that direction. We'll show a picture of what that would look like. Oh, geez. So here we don't have any attractant or any repellent. We're just kind of randomly moving around. It allows us to perceive our environment. So we could pick up on chemical, cues if they did exist. But here we might run, tumble, run, tumble. But we picked up on some sort of chemical in our environment. So now we generally want to be moving in the direction of the arrow. And so they'll try to control those runs and tumbles. And if they start getting further away or the signal is weaker, they'll tumble. Start getting where it's weaker, they'll tumble. And they'll try to keep in the general direction towards the positive chemical or away from the negative, right? So that's how they're going to respond. So it's not super controlled, but they're moving in a general direction overall. Whereas you can start to imagine that with our eukaryotic cells that can move in a whip-like fashion, they can literally control the directions that they're moving. They'll also respond to chemicals, but they can control that a lot more specifically. So bacteria are limited by the rumble thing with their flagella, but it does allow them to move quite effectively. All right, let's talk about how this works. So we're going to talk about the pilus and the fimbria. So let's just go on to fimbria. So fimbria is single, fimbriae is multiple. These are little bristle-like, kind of almost like hairs that are much, much shorter than the flagella, which are our long, long rotating guys. We have tiny little bristles on the outside of these. These are just for adhesion. They allow the cell to stick to different surfaces. The example that's getting us in the slide is E. coli. E. coli has fimbriae that allow for it to attach to your cell surfaces. And some types of E. coli, the diffusely adherent strains of E. coli, these guys are actually got such specific and capable fimbriae. They can be very able to attach to your cells in your, even in your urinary tract. So some bacteria in your gut, they can adhere to your gut cells, but the flushing action of your urine, they can't withstand. But certain strains of E. coli that we call diffusely adherent, their fimbriae allow them to attach inside of your urinary tract. And you might be thinking, well, that would suck to get E. coli for a urinary tract infection. Probably more than 80, it's definitely at least 80. Between 80 to 90% of urinary tract infections are caused by E. coli from your own butt. Okay, that's why we as women advocate wipe from front to back, not from back to front, because you don't want to get your E. coli where it don't belong. Most people carry E. coli in their gut just naturally. Very, very common part of your gut microbiome. So you want to keep it out of where it doesn't belong. We'll talk about how introducing microbes that belong in other places can cause infection. Most of us are aware that that's going to happen. We'll talk about how that works later on in other chapters. But this helps out with that stuff. So because they're able to have fimbriae that allow them to attach within your urinary tract, they can cause urinary tract infections. All right, pili, or the pilus is the single, so pili is single. These guys are made of protein called pilin. They're used in conjugation, and yeah, thinking like conjugal visits in prisons. Yes, I am talking about that actually. So we also call this the sex pilus or sex pili. So what these guys do is they form they can be used for attaching to surfaces, but more commonly, the most important function that they have, they can form these channels between cells. So they're still single cell organisms, but let's say I pick up a gene from some other cell or just from my environment, where I get a mutation that allows me to be antibiotic resistant. I can make copies of that gene, build a sex pilus to the cell near me, and send a copy over to my buddy over there. And they can transmit genes that way. So when we talk about conjugation, and we think about humans, sex, you can think of sex as being DNA transfer, swapping DNA, right? Same thing with the bacteria, they can swap DNA through the sex pilus. It's the closest thing that they're going to get to anything that even remotely resembles sexual reproduction. So they're not actually truly reproducing the whole cells this way, but they can help propagate extra genes that can help with survival and things. Not all cells can make the sex pilus, only certain species and strains can do it, but the ones that can, it's a useful thing to have. All right, so let's move on to the coatings that these cells can have. Bacteria can make It's basically a sugar. Sometimes it has protein in it, but it's very important that we remember that it's based on sugar. And it's kind of in the name. When you think of glyco, we're probably thinking of sugar, like glucoses and things like that. So glyco means sugar. Calyx just talks about coating. That's what calyx means, like outer coating. Okay. So the glycocalyx is like a sugary coating. And we have two different forms that you can have, the slime layer versus the capsule. The slime layer is more loosely associated, it's more watery, and it protects from loss of water and nutrients, and it also is helpful in like adhesion. And then we have a different version called the capsule, which is more dense and more tightly bound around the cell. And it's easier to visualize it typically. So let's look at a picture of that. Here we have a negative stain staining our background and a positive stain staining our cells. You see this white halo around the cell here? This is the area where the sugar coating is. And it didn't stain from either of these stains, so it's colorless. So this halo area represents that capsule. So all of these cells here, they all have capsules. If it didn't have a capsule, it would look like this. You see how that's cell color straight butted up against the blue background? That's not an artifact of staining. This would be a cell that does not have a capsule. That's the difference. So you can clearly see the capsule, and we'll be doing a capsule stain. You'd be surprised. Most cells, actually, most bacteria, especially our pathogenic ones, do make capsules. Okay. So here we see a difference here. We can actually, in some certain strains of bacteria, they might not always make a capsule, but only make it in response to certain situations. So here we have a bacteria that normally it might not make a capsule, but we've added or treated it in some way. Now it's making a capsule, and look at the difference. You can see it on the plate, that sliminess. So that capsule, that sugary coating, it literally makes it have that stickiness to it. So what's the use of that? Yes, it's very useful for adhering, so obviously sticking to surfaces, and that makes sense. If you had honey, just think of it as a coating like honey. It helps with sticking to surfaces, but it also is very useful for the bacteria. They'll coat themselves with these sticky coatings, and then it's hard for our white blood cells that are doing our immune functions and helping fight against, pathogens and other foreign things. Those white blood cells that help fight against those things are called phagocytes. Phago means eat, and cyte means cell. So phagocytic refers to the ability of our white blood cells to eat up things, foreign things. It could be dead cells of our own cells or debris, pathogens. I'm glad that happened. Okay, is it working yet? Hold on. Video, share this, share small. All right. So touchy, I didn't even touch it. All right. Anyways, the phagocytics, that's why I have bolded here. So that means cells that are eating up debris and other bad guys. Okay. And that's what our white blood cells are there for. We'll talk about how all that works when we get to immunology in chapters 15 and 16. But anyways, our capsules, that sticky sugary coating, think of it as being like a layer of honey that these guys are wearing. And it's hard for our white blood cells, our phagocytes, to grab onto them because of that sticky coating. Now we have ways to help us get around that. We'll talk about that when we get into it in those later chapters. But it's not 100% effective. And this is actually a pretty good way of pathogens avoiding being eaten up by our immune system. And like I already told you guys, most pathogens make these. So, all right. And then this is so they're good for adhering to things. They're good for avoiding being eaten up by our immune system. And they're also good for helping out with biofilms. And that makes sense. When we talk about helping with adhesion, helping cells stick to surfaces to start the biofilms, and helping cells stick to other cells. And you have this nice sticky coating that's holding all of it together. That's really what helps the plaque form on the surface of your teeth. And that helps it all stick there and makes it hard for you to get rid of it. This is part of why flossing is so important, in case you didn't know. Gets in between your teeth where your brush can't get to it, so that you don't build up any biofilm in between your teeth and then cause tooth decay. Because you guys, I'm sure you have a basic understanding of fermentation and byproducts that are made in fermentation. Because when we can buy kimchi and things like that, they have acid in them because the bacteria made the acid. Well, the bacteria on your teeth are making acid too, and that's just going to eat through your enamel the longer they sit there. And so that's why brushing and flossing is so important. You break up those biofilms so that they don't sit there and make acid that eats through your enamel, causing tooth decay. And that's why tooth decay or dental caries is the #1 infectious disease in humans. It's just so hard to control all of that. They hide out under your gums and in between your teeth. All right, moving on. The cell envelope. This is a term that refers to the entire outer layer of those cells. And I mean, everything that's on the outside. So we're talking about the cytoplasmic membrane, or the membrane that holds in the cytoplasm. You don't know what cytoplasm is? Cyto means cell, plasma, just think of like plasma. It's the goo that's inside of your cell that all the stuff is happening inside of. It's usually water-based, and there's like sugars and proteins and all of the structures of the cell in there. So it's mostly water, but the cytoplasm has to be held in by something. And the thing that is holding it in, that's contacting the cytoplasm, we call it the cytoplasmic membrane. And it's a membrane just like what we've been learning about. It's a lipid bilayer. It's got phospholipids all bound around it. So we've got nice negatively charged heads on the outside and those hydrophobic tails hidden on the inside. And that makes it weird for things to try to cross across. And that's what holds everything in, and keeps what's out. That's the membranes. Cytoplasmic membrane is the one that's going to be on the inside holding all the goo. All right. That's the basis of what we need for a cell. Bacteria typically also have a cell wall. We'll talk about what it's made of and how it works in a moment. Some bacteria might also have another outer membrane. So they might have two membranes. Let's talk about that. So the whole thing altogether is the cell envelope. And we often need to tell the difference between bacteria based on that cell envelope. There's two major groups of bacteria, the gram-positives and the gram-negatives. And it's literally based on how you react to the gram stain, which we'll talk about how that works. The gram stain is a staining procedure that when we go through the steps of it, we'll talk about how each step works and what it's for, that after you go through that procedure, if you stain purple versus if you stain red, you're gram-positive, positive P, purple. Negative, to me, red is a negative color, like stop at a stop sign or something. So negative, red. So based on whether you're purple or red, we define you as gram-positive or gram-negative. That's what really defines a gram-positive and a gram-negative. But why are we staining purple versus red? And that's usually dictated by that cell envelope. So whether you have a big, thick cell wall or a tiny, thin cell wall, but an extra membrane, those features are what are going to be able to define if you are staying purple or changing to red. Let's talk about how that works in a moment. So first, let's look at the structure of our cell envelope. We can, in gram-positives, so we have this mostly purple guy. And this guy over here, we'll call this the red guy. Gram-positives staining purple. We have this thick, where is it? It's over here. This thick purple layer here, this is our cell wall. Cell walls are made up of a molecule called peptidoglycan. And in gram-positives, that peptidoglycan cell wall is so thick. And we're talking 10 to 30 times thicker than the gram-negative layer of it. So that thick cell wall is actually usually what gives that cell its shape and its sturdiness. It's very sturdy. It's built like a scaffolding, and I'm going to show you how that works in just a moment. But this scaffolding, this thick scaffolding of sugars and peptides, and it's in the name, peptidoglycan, it tells you what it is. Peptido, you've got peptide bridges linking together glycan chains, or groups of sugars. They're long chains of sugars held together in the scaffolding by peptides or amino acid chains, right? That's all it is. So scaffolding built of that. Really thick layer on our gram-positives, so thick and sturdy. We'll actually attach our membrane to that pretty well. So that if I pop a cell that has that thick wall like that, nice strong wall, and I pop it into salt. What happens to cells typically when I put them in salt? They'll shrink, right? They'll shrivel. And all that water in the cell will be lost out into the salt. These guys might lose that water, but because they're attached to that big, sturdy, strong cell wall, they'll maintain their shape. So it won't kill them as easily. These guys are pretty well adapted to salty environments. So most of the cells, the bacteria that live on your skin, they're gram-positive. You have salt on your skin. They're adapted to live in that salty environment. The gram-negatives, on the other hand, they have a very thin wall. So they're going to be more susceptible to lysis from the salt concentrations. Okay, they're going to be flimsier in that regard. But they're also going to have more flexible cell wall. And they're going to be better built for other environments. We'll kind of introduce that later. But you can start to see the variability here. You're going to have thick, sturdy, supportive cell wall, or you might have the flexibility of the gram-negative guys. So one other thing we can talk about with our gram positives, because they have this huge scaffolding that's making up their cell wall, they need a little help maintaining that, especially if they have need to repair it from damage. And they also need help if they're like duplicating their cells. And I mean, these guys are always reproducing, and I mean fast. I mean, we can have a new cell made within a couple of hours. So every time that we're like making a new cell, we need to rebuild this freaking scaffolding. So we have molecules that help out with that. And this is lipoteichoic and teichoic acid. So TEICHOIC. That's only found in our gram positives, and that's to help out with that thick cell wall peptidoglycan scaffolding. All right, the gram negative guys. So they have a thin layer. And like I said, this could even be just one layer of those sugar and protein scaffoldings. It might not even have multiple layers. Very, very thin cell wall, but it is there typically, okay? And then they have this outer membrane. That outer membrane kind of compensates for the fact that they don't have the cell wall as far as maintaining their cell structure and things like that. But it also gives them another functionality for controlling things that are moving in and out of the cell, and another layer that they can put more functional proteins in and things like that. One of the molecules that they have in their outer membrane is something called lipopolysaccharide, or LPS. This is like the chemical name for it, the sciencey name for it, right? Lipopolysaccharide. And that also tells you what it is. Lipo being, we have a hydrophobic carbon hydrogen tails, right? Lipids. And polysaccharide, we have sugar units attached to it, right? So that tells you what that's made of. So this fatty chain with the sugars on it, LPS, lipopolysaccharide, we also call it another name in medicine, and that is endotoxin, which you may or may not have heard of. I promise I can spell. It's just really hard to write with this. Okay, so that's endotoxin. Good. Definitely what I wanted to do. Please. There we go. Okay, that's better. All right, so endotoxin. Endo means inside. Toxin means kind of what you would think it was. You think a toxin is causes damage or death to cells. Okay, so it's causing damage or death to cells. So endo, it's in because it's part of the bacterial cells. They're not secreting it out. It's just part of their outer membrane. So it's in them. Only gram negatives have LPS because they have the outer membrane. And this is part of the outer membrane. So LPS, lipopolysaccharide, AKA endotoxin. When your body sees endotoxin in it, it immediately kicks into inflammation gear. And this can be such a huge effect that if your body sees endotoxin in your bloodstream, you can go into toxic shock very, very quickly. So this is an indicator automatically to your immune system without having to make any specific response to that bacterial strain. Just the fact that it is gram negative and in your body will see it as foreign automatically. So that's another important thing. We will bring up LPS and endotoxin a lot. So that is something you do want to be familiar with. All right. So peptidoglycan, most bacteria have cell walls, and most of the cell walls are going to be made of peptidoglycan. So what is it? Again, mostly going to be repeating sugar units, which they have here as G and M. You don't need to know what this stands for, and you don't need to be able to draw it or recognize it or anything. But just to clarify, it's alternating N-acetylglucosamine and N-acetylmuramic acid, okay? So that's why they have G and M alternating, okay? So that's the sugars that are linked together, long, long chains of sugars. And then this green in the middle, these are our peptide crosslinks. And they're just like bridging together those long chains. So we have long sugar chains, sugar chain, sugar chain, sugar chain. And then bridging across those sugar chains, you would have these peptide linkages. And then this would be like multiple layers thick, you could imagine. And then that is basically what is going to be building up that scaffolding. So you start to imagine how this would be built. And so that's our peptidoglycan cell wall. You don't need to know what the amino acids are, but down here it does kind of delineate those amino acids that are involved in that. So that's our peptide chain linking together those sugar chains, peptidoglycan. So at least know that it's sugars and cross-linked by peptides. I won't ask you about the differences. I won't ask you about N-acetyl muriatic acid. Anyway, so moving on, you get the idea of that scaffolding. Very, very thick, 20 to 80 nanometers thick for our gram-positive, supported and maintained by the teichoic and lipoteichoic acids. In the gram-negatives, they have a thin layer of that cell wall, and we have on the outside of that another membrane. It's just another lipid bilayer, but that lipid bilayer is studded with these lipopolysaccharides. So these are less rigid, but they have more flexibility. It just depends on what you need as a cell. Now, if you are a gram-positive bacteria, you are always gram-positive. You can't switch. If you're gram-negative, you're always gram-negative. You can't switch. It's a characteristic. It's the actual bread and butter of all bacteriology. If I start talking about bacteria, most people who understand about microbiology and start understanding about bacteria, you're automatically going to think, is that gram-positive or gram-negative? It is that important for understanding kind of the functionality and relating between these. Yes?
Do you ever have any where there's both on there and then like with the different types?
Yes.
Multiple different types?
Yes. So if you did your gram stains properly, then 99.99% of the time, and for our purposes, if you have two different stainings, you have some cells that appear gram-positive and some that appear gram-negative. If for all of our purposes, you see that happen, it's going to be two different species. So one that is gram-positive and one that is gram-negative. So if you were to stain a sample from a patient and you're confident that this represents what's causing their illness, and you've got gram-positive, you got gram-negative, that means they're probably having an infection that might have two organisms involved. That can happen, unfortunately. Another thing that we see commonly would be if we were, a very good example, would be if you have a patient who is septic. And so you draw blood cultures, okay? If you were not good about cleaning your patient's arm before you took your blood cultures, or it wasn't sterilized, or you didn't maintain sterilized area for the IV, or how you were connecting your tubes to the IV, or anything like that, then when you collect your blood cultures, you might be collecting the bacteria in their blood, which could be gram-negative, for example. You can become septic with things like E. coli and things like that. But the skin surface bacteria, which could be gram-positive, could contaminate your sample, and now you've got blood cultures that have positive and negative in it. But that might not be because the patient's infected with the positive, but that you contaminated your sample. It's actually very common in people who aren't being careful when they're drawing their blood cultures. So that can be an issue as well. Something to consider whenever you guys get into nursing, you will be dealing with that sort of stuff. Even if you're going to vet, you'll be dealing with that sort of stuff with the animals. It's harder with animals because you can tell them to hold still as much as you like, but they aren't going to listen. So yeah, not that people always do, right? But anyways, moving on. So that was a great question. And if you guys have been looking at the critical thinking questions that I posted, there's a question like that on the critical thinking one as well. The whole point of the critical thinking questions is to give you data and you'll be able to pick out from the data what's important for the question. So sometimes the question is actually a lot simpler than what you really think it is. It might just be asking, how would you group this organism? And then you just have to pick the group that it belongs to, but it gives you all this other info and a setting in it, because realistically, that's what you're going to be doing when you guys get into working in the healthcare fields, right? So again, moving on, lipopolysaccharide, again, I'm not going to make you draw it or recognize it in a picture or anything, but this is kind of how it's set up. We have these long chains of fatty acids, that carbon hydrogen chains down here, lipid portion, and then up here we have our sugar portions, okay? Again, lipopolysaccharide, also known as endotoxin, it triggers fevers, what we call a pyrogen. Pyrode being fire, so it raises your body temperature immediately. Your body, it doesn't cause the body temperature, but your body sees it and says, time for fever. And it can also lead to shock. All right, moving on to non-typical cell walls. Some bacteria have weird cell walls, and some of them don't have a cell wall. So we'll talk about the major group of the weird first, and then the ones that don't have a cell wall next. So this first group is the group that has mycolic acid on the cell wall. So normally we think of our gram positives as having that thick layer of peptidoglycan, and then you have your membrane underneath it that encloses the goo. With these guys, they would still be technically gram positive, they'll stain gram positive, but they have these waxes on the outside. So they are lipid based, they're going to be hydrophobic, but it's like extra molecule chains that are attached to the outside of their peptidoglycan walls, okay? And this molecule here is a wax And just like other fatty acids, it might have an acid aspect to it, but it's really a lipid, okay? And this is a wax, mycolic acid. It's a long chain fatty acid. It is very hydrophobic. It says it contributes to the pathogenicity. Pathogenicity means its ability to cause disease and infection. So how does it do that? It makes them resistant to chemicals and to dyes and to oxygen. It doesn't dry out as quickly and things like that. So we use a special stain for these guys that is lipid soluble, and you can imagine that it would be. Most cells have a negative charge on the outside. These guys have wax on the outside. So the fact that it has wax, we can stain that wax and put that special stain on it that stains wax. And if you had a mixture of bacteria, the ones that don't have the wax won't hold on to that stain. And the ones that do will hold on to it. So imagine that your patient has one of these species of bacteria. This is only going to be seen in Mycobacterium. Look, it already has myco in it, just like this does. Myco. Mycobacterium are nocardia. Nocardia is not a major contributor to disease, and so I will never test you over it. But you absolutely need to know about Mycobacterium. So Mycobacterium includes diseases like Mycobacterium tuberculosis, causes tuberculosis, and Mycobacterium leprae. There's other Mycobacterium that can contribute to disease, but those are some two biggies. So Mycobacterium leprae is the cause of leprosy. These guys are very slow growing. They are very difficult to culture, but hey, you might not even need to culture them. Imagine that you had a patient come in with a cough, and they're coughing up kind of bloody greenish sputum. You would have them cough into a cup and collect their sputum and send it to the lab, and all they have to do is stain it with that stain. They don't need to culture it or anything. They're just going to take that sputum, stain it. It takes 10 minutes, and I can tell you if it has that waxy bacteria in it. Mycolic acid, so acid-fast stains, what that stain procedure is called. We'll do that in lab number 4. All right, moving on. These are mycoplasmas. Again, these have myco also. So there's a warning there. These are different. These have myco also, but the thing that's important for this guy is the plasma. At least the other guy will tell you it's a bacterium with myco, which happens to be mycolic acid. This doesn't have bacteria in it. So what's the important word here for us? Plasma, okay? So the plasma part is what we're focusing on with these guys. These guys don't have a cell wall. And the reason I bring up plasma is because imagine you were walking with a cup full of somebody's plasma from their blood. Nice golden yellow liquid portion of your blood. If I come over, and it's water-based, come over with a cup, literally just a cup of plasma, and I spill it on the table, will it hold its shape? No, right? Neither will these guys. And that's why they are pleomorphic. They have that softer shape. They don't have any cell wall at all helping maintain their shape. So they are pleomorphic. They don't have a set cell shape. They just have random shapes of their cells. All right. So that's how I remember the difference personally. Plasma, if I pour it on the table, it's not going to hold the shape. Mycoplasmas won't hold their shape. That means the other myco, the mycobacterium, are the ones with the mycolic acid, the wax. All right, moving on. Maybe. All right, yes. What's the function of that cytoplasmic membrane? Well, we already talked about how it's supposed to hold in the goo and keep what's inside inside and what's outside outside. The ability of your membrane to do that is called selective permeability. Water molecules and other very small uncharged molecules can cross across that membrane freely, but anything else is going to be kept in or kept out. And so you might want to be able to bring things in and out of the cell outside of that, but you can control that with protein channels and things like that. We'll talk more about how diffusion and things like that work for the cell in chapter 9, but that's the point of the membrane, okay? To control what goes in and out of the cell and to keep what's in and what's out. Let's talk about how the Gram stain works. The Gram stain is a really important concept to get how and why each step is happening. So our first step is crystal violet. It's a stain that is purple violet. And what it does is it sticks in that scaffolding. So if you have that thick layer of scaffolding that we already talked about, it is built up, and like that. You can imagine sort of like a Plinko machine. Probably don't even want to talk about, but anyways, you drop a thing in and it'll catch stuff in it and it'll stay in there when it's a nice thick layer. However, if you're just like one layer of peptidoglycan, the stain might just kind of sit in there until you rinse it out, but it's not going to stay in there. It's not going to get caught in there. And that's the whole premise of how this stain is going to work. So the first thing you'll do is put the crystal violet on there. At first glance, both cells will be purple. okay? But we're just applying the stain. Then we're gonna add iodine. Iodine is something called a mordant. Just like brick mortar, it's gonna help hold those dye molecules in there if they are stuck in there properly. So these guys are gonna get cross-linked and stuck in there by the iodine mordant. These guys will not be affected. These guys won't stick. Not permanently. Oh my gosh, I wish I had a pen. Okay, so gram-negative won't stick, but we still haven't rinsed out the excess yet. It's still just chilling, okay? So they're still purple. Next, we're gonna add the alcohol. Alcohol is a decolorizer. Let's see if I can do this. Yeah, that's better. All right. So alcohol is our decolorizer. And so that's going to remove any unstuck of the, oh my gosh. This removes any crystal violet that didn't stick. You know what it says. All right, so that's pretty much what the alcohol is going to do. So here we can start to see now, if we were removing anything that didn't stick, that we're going to stay purple here because it's stuck. It got stuck in the Plinko machine, got stuck in the scaffolding. We add alcohol to our gram negatives, it's going to rinse out all of that crystal violet and it's going to become colorless. Oh my gosh, I just typed it, colorless. You guys love that sound already? We're going to be right next to it in a minute. All right, safranin is our next step. So you can see that if you were to look at this colorless guy in a microscope, you probably couldn't see him. So safranin, its whole purpose is just something called a counterstain. And this will make a lot more sense once we do lab 3. On lab 3, we're going to do the gram stain. You're going to see it in practice and how it works and everything like that. But this is the theory behind it. So this is why positives are purple. They hold on to the crystal violet, and negatives are red. They got rinsed out with the crystal violet, and they stain red because safranin is red. That's our counter stain. Make sense? Okay. All right. We already talked about the cytoplasm, which is the goo inside of the cells, just where everything is going on in it. Bacteria, chromosomes, they are DNA. They exist as a circle, one single circle. All of their DNA is just in one circle. Our genes are in multiple linear chromosomes. We'll talk about the difference in chapter 6, but these guys, it's important to remember, circle, it's only one. But they can also have accessory kind of DNA pieces. These also will exist in a circle, but they're much, much, much tinier. And they have non-essential genes on them, but genes that can help them survive better. So we're talking about antibiotic resistance, maybe the ability to make certain toxins or enzymes that help break things down. They can be on the plasmid. So it can still exist without the plasmid, but it will help give us extra genes. And these guys we can make copies of and shuttle across in our conjugation visits, okay, with our sex pilus. Ribosomes, I don't want to get into too much on what a ribosome is, but they're basically RNA, maybe a little protein in them, mostly RNA, and they are the machines that are going to help us make proteins. We're going to talk about their full function in chapter 6. But bacteria have one size, it's called 70S. Don't worry about what it means. They have one size, whereas eukaryotes have 80S, it's just a different size. And so it's one way you can tell the difference between them. Bacteria can also make something called endospores. These are just structures that a cell can make one endospore if it is capable of it. There's actually only 2 genera, or genuses, is another way of saying it, that are capable of making endospores. And this is Bacillus. So your name might be Bacillus anthracis, for example. That's Bacillus anthracis causes anthrax. Or Clostridium species. Things like Clostridium tetani causes tetanus. Okay, so these guys make endospores. These are hardy, resistant, completely inert, resting, dormant structures. They only make these when they're scarce of nutrients or water. So they'll make these structures. They have this outer layer called the cortex that they make that makes them resistant to harsh conditions. And I mean, they can just exist like that for decades, just waiting to be reawakened. It's usually reawakened by water or something, or nutrients. And so they have a whole life cycle that's involved. Let's go over it just briefly. Again, resistant to heating, to drying, to freezing, to radiation, and to certain chemicals. There are things that can kill these guys,
That was my question.
right?
Yeah.
Things like autoclaving and certain chemicals like or concentrations of like chlorines and other halogens like that. But basic things aren't going to get rid of this. For example, boiling doesn't get rid of an endospore. Boiling doesn't sterilize. You can boil to your little heart's content. You ain't going to sterilize. Spoiler alert, okay? Won't get rid of endospores. And these guys really are everywhere. It's in fact the whole reason behind like brewing and the steps we have to take to sterilize the equipment is because if we're not making certain pH levels that can affect the protein functions of Clostridium botulinum, then you're going to end up with your beer having botulism in it. If you're not sterilizing your cans that you're canning your foods in, then you are at risk for botulism because it's just freaking everywhere. It is. And so just most of the time we are trying to treat our foods and things like that to prevent it from being able to grow. So here is how these guys work. This is the cycle. The vegetative cell is the active multiplying version of it. In fact, any cell that is, metabolically active and dividing and multiplying, whether it can make an endospore or not, those are called vegetative cells. But if you are able to make endospore, each cell can only make one. So this is not a reproductive method. This is just for, survival. So you become something called a sporangium. That's the cell that's forming the endospore. And it'll make the endospore within it. And then the endospore will exist free outside of the sporangium. And when it gets activated by it'll go through a process of germination. And that germination is when we're going back into the vegetative state again. The sporulation, making the endospore, germination, coming back into vegetative, okay? It takes about 6 to 8 hours to make an endospore. It only takes 1.5 hours to wake up again. So you go out and you're playing outside in backyard of your grandparents' place, and you step on a nail. Clostridium species hate oxygen. They can't live in oxygen, but their endospores can survive it. You stick those endospores all the way up inside your foot from stepping on that nail.
More oxygen.
They don't have any more oxygen. Exactly. So now they can go back into vegetative form and start making the toxin associated with tetanus, which is called tetanospasmin. Tetanospasmin causes your muscles to seize up. They cannot relax. You don't just seize up your jaw. I want to be clear about that. You'll eventually seize up every muscle in your body, including your diaphragm, which is how ultimately you will, you will suffocate and die. Yes.
So like endospores are kind of just like dehydrated.
It's like
food in like a where you could just like
So
when it gets reintroduced to water, nutrients, it's alive again.
they'll just become alive again.
Okay.
And they have everything that they need to like rebuild the cell and its structures. It's sort of like protecting just the genome and the things it needs to turn that genome into the cell again.
Right.
In this like layer. So we have tetanus that'll do that. And we'll talk about botulism. And obviously botulism causes such relaxation of your muscles and you can't contract them. So it's the opposite, but they're both Clostridium. Okay, so we've got Clostridium, we've got anthrax. Obviously, they can be dried out and disseminated. We've seen that done literally to Congress. In 2002, somebody sent anthrax to Congress and it got disseminated there. And somebody tried to do bioterror. So these things, this is a serious thing. These guys can live for decades just chilling in the soil. And there's a lot of them. Anthrax, Bacillus anthracis, Clostridium tetani for tetanus, Clostridium perfringens causes gas gangrene. And we have Clostridium botulinum, botulism. We'll talk about even more of them later on. Archaea, just really quickly, these guys don't have true peptidoglycan. They have some other wall, and they have some other kind of weird membrane too. Archaea, really weird. They're real super weird, but they're still single cell, and they don't have a nucleus. These guys produce methane, or they live in extreme environments. All right. So methanogens, these are just organisms that produce methane. Methane is a bigger contributor to global warming than probably carbon dioxide is. In fact, you might think, well, cows produce methane. Yeah, the bacteria in the archaea in their guts produce methane. So not the cows, the bacteria in the archaea. We also have extreme halophiles. Halo means halogen. So here we're talking about guys that can survive high salt concentration. In fact, they can live in salt. So often these are going to be our archaea. Archaea can also survive extreme low temperatures. So we're talking about sub-freezing. They'll thrive in these temperatures, or above boiling. We have hyperthermophiles that can exist between 80 and 113 degrees Celsius. 100 degrees Celsius is boiling. So we're talking about, these are the guys that live in those volcanic vents in the deep seas and things like that. We can classify our bacteria based on their envelope. It's just one way we can say, oh, you are an eukaryote, you fall into one of these 4 groups, all right? I always start with the firmicutes. Firmicutes, firm. Why is it firm? Because it has a thick cell wall. So it is gram-positive. Easy start. All right, the next one I go to is, if that's our gram-positive, what's our gram-negative? It's the GRA one, okay? So gram-negative. Grassy means like thin-skinned, is what this really, literally the word means, thin-skinned. So you could look at it that way, but I just say gram, that's the one that isn't firm. So that's our gram-negatives. So they're not as firm. They don't have that thin cell wall. And then next, we have the Mendoza-cutes. I already told you about how Joey Mendoza was cute. Mendoza cute, that was a long time ago. Archaea, old, long time ago, right? So that's how I remember that. That was a long time ago. I was very archaic that I thought that Joey Mendoza was cute. You guys can use that to your advantage, okay? And then the Tenera-cutes. Tener literally is basically the same thing as tender. Not Tinder, but tender. So what's tender? It means very soft. So we have no cell wall for these guys. And that's how we classify them. Cool? Again, this is just memorization for these guys. All right, last. Oh, yeah. Well, you can look at different traits. Like I could say E. coli, right? We know we have E. coli in our gut, but we also know there's some E. coli that can cause very, very serious disease if it's present in your food. It's not the same exact strain as the E. coli in your gut. They are both E. coli, same species, but they are able to make a certain kind of toxin that leads to something that we call dysentery. Dysentery is not just something that happens on the Oregon Trail. Dysentery means bloody diarrhea. So if you have bloody diarrhea, the word for that is dysentery. And so strains of E. coli are able to make the toxin that leads to dysentery. Not all of them can do that. Obviously, the ones that are causing your UTIs, they're not dysentery causing ones.